So this lesson is on mutations. Your key question: what are mutations and why are they the ultimate source of new alleles and therefore genetic variation? Your target objective: students will be able to understand how beneficial mutations increase the fitness of the organisms that possess them. Critical attributes. Today I will learn as a student about mutations so I can recognize that inheritable genetic variation may result from errors occurring during replication. And this is during the S phase, synthesis phase of the cell cycle. I know I have it when I can make and defend a claim based on evidence that may lead to genetic variations. So to introduce mutations, we will first define what a mutation is. So a mutation is a change in DNA sequence and occurs in genes, which are segments of DNA. Mutations can be harmful, beneficial, or neutral. Um, the key to genetic variation and evolution ties into mutations, and different types of mutations are your points, insertions, deletions, and chromosomal mutations. So here I have an example of a mutation that has happened with this particular peptide chain. So this gene that codes for this specific peptide chain is on the chromosome 13, and normally between 1 to 2 steps is showing us a normal occurrence. So the normal DNA sequence is here. You have the normal amino acid sequence. So the code is read in 3 base sequences called codons, and then 1 codon codes for an amino acid. Now, a mutation could possibly happen to that chromosome 13. Well, it will result in an early stop. So when it's supposed to continue to code for that amino acid sequence, it actually stops. So it results in a shorter chromosome 13, and the effect of this would be a person that can no longer hear. So let's look at more of these types of mutations. So point mutations, one example is a substitution. So one base pair is replaced by another. So looking at this example, what's normal as ATG, we actually have ATC. Insertions and deletions, what they are is what they're doing. So insert, you are adding one or more base pair. Deletion, you are removing one or more base pair. And these two can result in a frame shift mutation. Your chromosomal mutations is more of a large scale change in chromosomes and that chromosomal structure. And like we've seen before in the picture there in the chromosome 13, and examples of that will be inversions, translocations, and duplications. So this is just a picture image of the 3 different types of mutations that we talked about previously. So that's deletion, insertion, and substitution. Looking at the cell nucleus where we would find DNA. This is the normal DNA sequence, and under these 3 occurrences, this is what you would see. Okay, so your causes of mutations, these are spontaneous errors during DNA replication. So during DNA replication, we could possibly have a misprint there, and that's what we just seen before. Also, mutations can be caused due to environmental factors. So we call those mutagens. So that's exposure to long exposure to sun, X-rays, certain chemicals, and viral infections. And also, they can be inherited mutations from parents. Okay. And your genetics, again, so your causes of mutations are your genetics, environment, and epigenetics. So that DNA methylation, histone methylation, so the process of expanding and exposing the DNA versus condensing it. We can have a mutation happen there based on the environment and, of course, what's inherited. So the effects of mutations, your solid mutations have no effect. So you have no change in the amino acid sequence. So a mutation did indeed happen, but we do not have an effect. Your missense mutations have a change in one amino acid. Your nonsense mutations have a premature stop codon, and frame shift mutations will alter the reading frame altogether. And this can lead to genetic disorders, cancers, and evolutionary adaptation. So this is an example of silent, nonsense, and missense. So your silent mutation, it is what it is. What that word means, silent, quiet, no effect. You can see even though a mutation happened, we still ended up with the same amino acid that we want. Nonsense mutation actually stops the coding early. So a code for stop will happen when we're supposed to originally have this amino acid here. And our sense mutation will produce an entirely different amino acid. So let's recall some causes of mutations. All right, so let's get into beneficial mutation. So one beneficial mutation, it's a positive one because it helps the plants grow. So there's a mutation in plants that have developed over time where they are naturally resistant to bacterial and fungal infections. Some other beneficial mutations would be lactose intolerance in humans. Lactose tolerance in humans, not intolerance, lactose tolerance. So they're able to take in dairy without having the effects of a person who is lactose intolerant. Antibiotic resistance in bacteria. So bacteria benefits from this because they are not killed off by antibiotics. Now, it's good for the bacteria, but not good for the host that the bacteria is in, right? So, for example, if you're a human, right, and you experience a bacterial infection, harmful bacteria that's taken over your body, um, you wouldn't want antibiotic resistant bacteria in your body system because it will be hard to treat that bacteria because they are naturally resistant to the commonly used antibiotics used to treat bacteria. So, like I said, it benefits bacteria, the population of bacteria, but it doesn't benefit the host. And also pesticide resistance in insects. So let's get into our harmful mutations. So this can cause genetic disorders. Examples would be sickle cell anemia. This is where we have an irregular shape of the human blood cell. The red blood cell is responsible for transporting oxygen and nutrients to the body. So when you have an irregular shape, it's very difficult for oxygen to attach to that red blood cell. So that, of course, would be a negative point because oxygen is used to transport and support the body systems. Cystic fibrosis is another genetic disorder as well as Huntington's disease. So just to recall, I know we just spoke on harmful mutations, but try to see if you can remember in what ways mutations can be beneficial. Now, detecting mutations. So you have geneticists, scientists who are designed to look at the genetic history or the genetic makeup of a living organism. So say for instance, you are married and you're ready to have children, but you are not quite familiar about your own genetic history and your partner. So some people will actually make an appointment with a geneticist and actually get pictures of a karyotype. And a karyotype is just simply a picture of your chromosomes, of your 23 homologous paired up chromosomes. And of course, if we count this out, it will equal 46 to make up that fully diploid organism. But keep in mind, one of these chromosomes, or technically, one of the chromosomes came from mom, the other chromosomes came from dad, okay? So that's why you have 23 homologous paired chromosomes seen on a karyotype. Now, the 23rd chromosome is the detection of the sex. Okay, so you should be able to look at a karyotype and determine the sex of that person. Okay, even without the labels here. So, because females are represented by two X's, you would see them same length, okay, same size there. But for a male, it will be obvious because we will have one long chromosome and then one short one. All right. So the rest of these, 1 through 22, are considered your somatic or your autosomes. So those are body chromosomes, and these are your sex chromosomes. Okay. So this is what a normal karyotype will look like. You will have pairs of each of those chromosomes. They will be, similar in length, size, shape. So this is how you can tell that this is a normal karyotype, and you should be able to tell the sex as well. Versus looking at an abnormal karyotype. you see here. But before we look at and dive into your Down syndrome karyotype, let's see if you guys can look here and tell me what the sex of the abnormal karyotype is. Just compare it to the normal karyotype, and you should put female because one, it gave it away. But if those two X's were not there, you should have still felt comfortable with saying female because these chromosomes are the same length. Now, if this one was a bit shorter, obviously shorter length, then that would be indicative of a male karyotype. But we're looking at a female with Down syndrome. And if you can compare the 21 chromosome, you're only supposed to have a pair. You're not supposed to have three. So a mutation drove that, right? So we have three chromosomes for that 21 pair, the 21st pair, which causes Down syndrome. Also, mutations can be treated. So scientists are working on treating these mutations by gene therapy. So they use a viral injection. So it'll act like a virus, but what's inside of it, of that virus viral capsule, is something to correct that mutation. CRISPR-Cas9 is very popular. It actually edits the gene, so the effective gene on the DNA sequence, it's able to cut that out without affecting the rest of the DNA sequences. Okay, so this is a sample leap challenge question. Heterochromia is when a human or animal has two different eye colors. Heterochromia, I'm sorry, I said heterochromia. It's heterochromia. Can have many different causes. So the cause of heterochromia determines its type and if it is heritable or not. So the person in the image has this mutation, right? Has this disease. And this occurred during mitosis, and it is only found in those chromosomes from 1 to 22. So the somatic body chromosomes, not the sex cells, okay? So with that being said, your job is to choose the best that describe why the person in the passive image has two eye colors. So just looking at the answer choices, you would have to do process of elimination. And because it says the best, I'm not 100% comfortable with these type of questions because technically you may have another right answer, but it's not as correct as the one that they're expecting you to choose. So with that, you definitely want to use process elimination, that process to help you narrow down your choices. So you have to determine if this a mutation that only affects one eye or a mutation that affects two eyes, both of your eyes, or is it a mutation or is it a temporary change or a permanent change? So unfortunately, it's definitely a permanent change. So that narrows out A and D. So out of B and C, what do you think would be the correct answer? So if you chose B, you are correct. So a mutation in a cell of one eye altered the DNA to express a permanent change. All right, so to conclude, mutations are fundamental to genetic diversity. Understanding mutations is crucial for medical advancements, evolutionary biology, biotechnology, and ongoing research continuing to reveal the complex role of mutations in life. So to prepare you for your mastery check, you should be able to recall everything that's on these 5 bullets. So if you're working in groups, test yourselves, ask each other questions without looking at the notes to help better prepare you to score 75% or higher. So what is a mutation and how does it occur? Name and describe 3 types of mutations. How can mutations be both harmful and beneficial? What are some environmental factors that can cause mutations, and how are mutations detected, and why is this important in medicine? So you should be able to recall all of that with confidence. And if you feel like you're confident, then go ahead and get my attention and take your mastery check. So to finalize this lesson, you should feel confident to again answer the key question at the start of the lesson. So what are mutations and why are they the ultimate source of new alleles and therefore genetic variation?